Hi everyone, welcome to the 25th Keio Medical Science Prize lecture. So I am Haruhiko Shiomi, chairperson of the Keio Medical Science Prize Committee. So I am very pleased to announce this year, 2020, the Keio Medical Science Prize was awarded jointly to the Dr. Aviv Regev and Dr. Atsushi Miyawaki, recognizing their creativity and ingenuity in developing a number of cell imaging techniques and tools that have greatly contributed to basic biology and medical sciences. I am also very pleased to learn that both Dr. Regev and Dr. Miyawaki image cells and tissues just in different but very complementary ways. So today we have a Keio Medical Science Prize lecture by Dr. Aviv Regev. So Dr. Regev has been an essential driver of the rise of an entirely new research field of biology in the last few years, so namely single-cell genomics. Previously, researchers profiled gene expression in tissues in bulk, analyzing many cells in a single experiment. In order to be able to study the behavior and gene expression patterns of individual cells, including rare populations that are lost in bulk sequencing, Dr. Regev built a power suite of single-cell sequencing tools. So single-cell genomics allows us to examine biological systems and the tissues of the human body in health and disease in an unprecedented detail. So in a short few years, single-cell analysis has revolutionized our understanding of the composition of human body. So these advances have largely been possible due to Dr. Regev's development and deployment of new technologies and computational methods across a variety of systems. Beyond her own work, Dr. Regev had the idea to build the Human Cell Atlas in 2014, when single-cell sequencing technologies were still in their infancy. So since then, she and her colleagues have convinced international community of more than 2,000 scientists dedicated to charting the cell types of the human body as a reference map to study health and disease. So before her lecture today, we have two special guests who will be congratulating Dr. Regev. So first, it is my great honor to introduce Her Excellency Mrs. Yafa Ben Ali, ambassador of the State of Israel to Japan. So Your Excellency, Mrs. Yafa Ben Ali, would you please give us the honor of addressing us now?
Well, thank you very much. I would like to, be very short, obviously, but I am expressing here the great admiration for creative minds and scientific development at the same time. Definitely, Dr. Aviv Regev brings us a lot of honor, and we are very proud of her achievement, being the second woman to win such an important prize, and as well as the first Israeli to be awarded with the Medical Science Prize. I believe that these days, at the time that we are facing the very critical pandemic, the COVID-19, with a lot of uncertainty, finding the right roadmap to find solution in the medical field, I think it's extremely important and even representative of the importance of global scientific collaboration. That particular development, and I'm not expert in biology, but as far as I can understand, will bring us, or has been already bringing us, a lot of hope for health of people around the world. So I wish to conclude in saying thank you very much. Thank you, the Keio University, as well as thank you, the two honored scientists, for the breakthrough they have been bringing to this world, especially at this very time. So congratulations once again, and let us hear the great news as it is told. Thank you.
So thank you very much for the very warm greeting. So next, we have the pleasure of being joined by Joseph Young, charged Affairs at Interim, U.S. Embassy, Tokyo. So, Mr. Young, would you please give us the honor of addressing us now?
Sure. Your Excellency, Ambassador Ben-Ari, thank you for your wonderful remarks. Professor Shiomi and Professor Kubota, thank you for hosting today's lecture and for your selection of two very impressive laureates, Dr. Miyawaki and Dr. Regev. The importance and creative and innovative medical advances like those advanced by the Keio Medical Science Prize has become clear to the global population like never before in 2020. On behalf of U.S. Embassy Tokyo and the American people, it's my great honor to have the opportunity to congratulate Dr. Aviv Regev, the recipient of the 25th Keio Medical Science Prize. This prestigious prize recognizes Dr. Regev's outstanding contributions to advance single-cell genomics and acknowledges her work to foster a spirit of partnership among the international research community. Dr. Regev's novel techniques to profile the human body at single-cell resolution will play a vital role in the future of diagnosis and therapy. The International Human Cell Atlas Project, for which Dr. Regev serves as organizing committee co-chair, brought together an international community of scientists to create a comprehensive reference map of human cells as a foundation for understanding human health. Dr. Regev's visionary leadership and technical expertise is indispensable to the project's members from more than 1,000 research institutes and 70 countries. I extend my sincerest congratulations to Dr. Regev. Her contributions to medical science are admired, respected, and appreciated. Though the novel coronavirus prevents us from being together in person for her lecture, the silver lining is that her virtual lecture today will be able to reach experienced and aspiring scientists globally. Role models like Dr. Regev will inspire the next generation of young women and men to become scientists for the betterment of humankind. We look forward to her continued success and her future endeavors. Thanks very much.
Thank you so much for the greeting. So now it is my great pleasure to introduce Dr. Aviv Regev. So would you please speak to us and share your ideas, thoughts, and perspective with us?
Thank you so much. Thank you for the selection committee, and thank you also so much to Ambassador Ben-Ari and to Charge d'Affaires Young for their very kind words. As an Israeli-American, both of them touch me very much. I'm extremely honored to receive this award, the Keio Award, and for the general recognition that the award conveys to the field of single-cell genomics. So I'm going to start with my disclosures. I'm a co-founder and equity holder in Celsius Therapeutics and Immunitas, and until July 31st, I was an SAB member with cash or equity compensation in Thermo Fisher, Neogene, and Syros. And from August 1st, I'm an employee of Genentech, and my lab is actually moving there. But the work that I will show today was done in my lab at MIT, the Broad Institute, and the Howard Hughes Medical Institute. And I'm going to start with cells, which are the basic unit of life. Each of us is made of about 37 trillion cells, which come in many different types, like neurons, immune cells, muscle cells, and fat cells. And at first glance, you might think that every cell in your body is the same, because after all, it has the same DNA, and so the same set of instructions. But in fact, cell comes in many different structures and locations, and they perform different functions in our body. And so we must ask ourselves, how come? Well, even though every cell in our body has the same DNA instructions, which part of the instructions they actually use and to what extent, that is how much RNA and protein a cell will actually make from its genes, depends on what it needs. And so imagine that there's just 4 genes, A, B, C, and D, and there are just 3 kinds of cells, neurons, immune cells, and muscle cells. These cells need to do different things. The neurons process signals, immune cells respond to pathogens, muscle cells contract. And so to do this, they need proteins that are coded by different genes. So for example, cell 1 may need some of A and less of C. So it's going to express multiple copies of RNA from gene A, fewer from C, and none of B or D. And then, I'm going to label how much it expresses of A, B, and C by these colors of blue. So the darker of blue, the more the gene is expressed. Now, cell 2 may express each of A, B, and C, but none of D, and I'm going to color it by these lighter blues. And finally, cell 3 might express high levels of B and D. Okay. And so the pattern of how much each gene is expressed is what we call an expression profile. And this profile is like the calling card of the cell. Because if we know what the cell uses, then we know what the cell is. Cells that are similar to each other should have similar calling cards. All the neurons are going to express similar genes. Immune cells express other similar genes, and so on. Now, why does it all matter? The fact that all cells in our body have the same instructions, but they use different parts of them, it has critical implications when we start to think about the genetic causes of disease. And so if there's a genetic variant that confers risk for disease, all the cells in our body are going to carry the variant. But the disease is only going to manifest in those cell types that use or express this gene. And so knowing our cells is essential for understanding how the genes that cause disease act. So in terms of those disease risk genes, in the past 15 years, another area of biology, human genetics, has really brought the world 100,000 regions in the genome that are associated with a range of diseases, from inflammatory bowel disease to schizophrenia. And this was a remarkable achievement and an outcome of the Human Genome Project. But if you behold this kind of image now, it hides one of our biggest new challenges for biology. How do we take these 100,000 variants and we understand what each of them does in the disease? And in particular for this, we need to know the cells in which they act. And the only problem that we have is that we don't really know how many different cells are there and who they are. So if you would open, say, Wikipedia, it would tell you that there are maybe 300 major cell types. But if you ask a neuroscientist, they would tell you that just in your retina, there's more than 100 subtypes of neurons, each of them doing something different. And so biologists have always been on a quest to identify all the cells, ever since they were first discovered under the microscope in the 1600s, and especially for the past 150 years or so. And it's been an endeavor that was always driven by new technologies, first the microscope and then the latest probes. But as a result of this, we actually ended up with many different ways of defining cells. And what we really need is a map or an atlas that has clear coordinates and that would let us know all of our cells and what they do. And so you might ask yourself, what might be the coordinates of these maps? Well, the coordinates are going to be the level of expression of these different genes. So how would we think about a map like this? Well, imagine that there's only 3 genes, each of them captured by one axis. And so, for example, this big cell in the middle might express a lot of gene 3 and very little of gene 1 and gene 2. And so the cell is going to be a point in 3-dimensional space. Our genome doesn't have only 3 genes. It has about 20,000 genes. And so you can imagine that there are 20,000 coordinates in the map. I can't show you 20,000 coordinates on a slide, but math and machine learning algorithms can actually handle a space like that. And they can see the map for us, and they can discover its patterns, as I will show you. And so again, this is all wonderful, but how would we get the information to put the cells in the map? Well, for a long time, we actually couldn't precisely do this until new lab methods came along that are called single-cell genomics. And so until recently, if I wanted to know the expression profile, how much is each gene expressed from the genome, I had to take hundreds of thousands or millions or even billions of cells, and I had to measure their average. And so if you think of each cell as a piece of fruit, we were basically generating the lab equivalent of a fruit smoothie. In the last few years, there's been this major breakthrough called single-cell genomics. In single-cell genomics, we measure the expression of genes in each cell separately, individually. So you can think of it as the lab equivalent of a fruit salad. And the very latest spatial technologies allow us to do these measurements when the cells are all organized together in the tissue, just like they are in the body. And so this we think of as the lab equivalent of the fruit tart. Today, I'm going to focus mostly on single-cell genomics. So I'll tell you a little bit about how it works. The technique that I'm going to focus on is called single-cell RNA-seq. It was the first one to be developed and applied at scale. And we start with a piece of tissue, which we dissociate, we separate to make it into a suspension of single cells. And then we capture each of these cells separately. We tag and isolate the RNA molecules, and then we can sequence the RNA on the sequencer. And this is going to tell us two things. One is the identity of the genes that they actually came from. And it's going to tell us how many copies of them were expressed. And because there's so many cells in the body, we actually needed a way to do this really quickly and really cheaply for every cell. So one approach that allows us to do this, the first one to be developed, is called DropSeq, which we developed together with Dave White's and Steve McCarroll's labs, and is shown in this movie. And the cells here get captured in these little tiny droplets inside tiny devices. And using these kinds of massively parallel methods, we can now study millions, first thousands and then tens of thousands and now millions of cells in one day. And this really led to this explosion in the number of cells that we can look at. And of course, the question that would come up is, what do we learn from this? And what I'm going to show you in this talk through a series of vignettes is that in the past several years, single-cell genomics has allowed us to shed light on the most fundamental aspects of the organization and function of cells and tissues, and that these, in turn, then gave us a new vocabulary for disease studies. My specific vignettes are going to mostly come from our most recent studies, but I especially want to pay tribute to our initial series of studies that spanned primary cells from the immune system, human tumors, and animal models, and only some of them I will actually highlight today. Because having this diversity of biological systems to examine really allowed us to bring single-cell genomics to the forefront. And so for my first story is going to be about discovering cells in our body that we didn't even know exist. And this is going to start with a mouse and a part of the airway known as the trachea. And so to know which cells make the lining of the airway, Dan Montoro, who's a grad student in Jared Jagopal's lab, working with Moshe Beaton and Adam Haber in my lab, profiled 40,000 cells that I'm showing here. Each cell is a dot, and the cells are colored by their major groupings or clusters. Now we can readily rediscover the 3 common and 3 rare subtypes of cells that we already knew exist in the airways, except that we found that there's also 2 subsets of goblet cells and a progenitor for neuroendocrine cells, 2 subsets for staph cells and a progenitor, and new specialized clap-like cells that we called hillocks. All of these things were new for us, but I won't get into them because they were at least similar to something that we've seen before. But in addition, there was this speckle of a subset here, which did not map to anything that we've seen before in the airways. And so we asked ourselves, what are these cells? And so, as their calling card, we identified which genes, which are in the rows, are uniquely expressed by these cells compared to all the other cells which are in the columns. And we see that they express the VATPA subunits and the transcription factor FOXC1, which are known to be expressed in cells in other species like frog and fish that are called ionocytes, as well as cells in the inner ear, the kidney, and the epididymis. These are all cells that regulate ion transport and fluid pH. And so we decided to call these cells the tracheal ionocytes. But we also had a major surprise because these cells also express a gene called CFTR, also known as the cystic fibrosis gene, the 3rd gene to be cloned by modern human genetics for a human disease. And this is a gene where the literature from 30 years has been telling us is expressed in another kind of cell, the ciliated cell. But our data shows that CFTR is actually expressed in the speckle of cell we didn't know about, known as the ionocyte, and not really in the much more abundant ciliated cells. And we validated this in multiple ways in situ. And because we first found this in the mouse, we then went and looked at hundreds of thousands of cells from human lungs. And again, we found these ionocytes, and we found that the cystic fibrosis gene, CFTR, is expressed in ionocytes and not in ciliated cells. This is important because as we develop therapies for CF, especially cellular therapies, we had better know the right cell type to target. Okay, so now let's think about where are cells located. Typically, we think that if we want to know where the cells are, we need to measure them in situ, in the tissue. But is there information that's actually embedded in the expression profile of dissociated cells that can enrich our knowledge? And so several years ago, Rahul Satija, when he was a postdoc in my lab, together with Jeff Farrell, when he was a postdoc in Alex Shear's lab, wanted to find a way to relate single cell profiles from dissociated embryos with spatial patterns that are measured in situ. And so we're going to begin with a little bit of information that we already have, legacy information. These are these in situ patterns for landmark genes X, Y, and Z. And from these together, we can form a reference map. Now imagine that we had one cell that expressed the red and the blue gene, but not the yellow. And so we could predict that it comes from this bin. And then we have another cell with the yellow and the blue, but not the red gene, and we can predict that it comes from this bin. And so we implemented this idea in an algorithm that we called SERA, and which allowed us to handle the noise in our data and does the mapping probabilistically. And so as our first test case, Jeff used embryos of the zebrafish from a particular stage called 50% epiboly, an early developmental phase in fish development, to show that SERA's predictions come within one bin in each of the axes of where he manually recorded that he plucked cells individually, which is basically Jeff's own confidence in his manual process. So now that we mapped our cells, we can use the mapping to infer the spatial expression of genes that were not in our reference map. Our reference map might only have a dozen genes or 30 genes, but the zebrafish genome has tens of thousands of genes, which we have not measured experimentally. So imagine that our mapped cells are here, and there's some gene X that's expressed at some level in each cell. We can now infer an in situ pattern for the gene in each bin by weighing its expression in each mapped cell with the probability of mapping that cell to the bin. And so here are 9 such predicted in situs, which Jeff then validated with an actual experiment in situ, showing that the predicted and the experimental in situs agreed really well. So this lets us map cells to their histological position, their position in the tissue, but can we get all the way back through to a full organ or the entire body? And so to do this kind of mapping, we developed a new algorithm we call Tangram, like the game, which solves this nonlinear optimization problem iteratively, similar to placing puzzle pieces. So we initiate by randomly distributing single cell RNA-seq or single nucleus RNA-seq profiles into a region of interest. Then we score the mapping by computing a loss function, and then we optimize the mapping with stochastic gradient descent iteratively. So of course, the key point is this Tangram loss function, which compares the mapping of our cells to the spatial experimental data by considering 3 terms: a density term, a gene expression term, and a cell number term. For the gene expression term, we compare the mapped expression to the spatially measured expression for any gene for which we have a measurement, both for each of the genes across the voxels and for each voxel across all of the genes. We also assess this density, the cell density term by the KL divergence between the mapped single cell RNA-seq and the spatial data. And now for the cell number term, imagine that our section only had 1,000 segmented cells, but our single cell RNA-seq dataset had 30,000 profiled cells or nuclei. We want to select just the best 1,000 cells that explain the data. And so for this, we also use a filter called F, which is convolved in all of the previous matrices. And finally, to relate to histology and anatomy, we also learned the model of anatomical depth, in this case for the brain, from an existing brain atlas, so that the point distance in the learned latent space represents the anatomical distance between the slices. We also learned the model of anatomical labels, again, in this case from the Allen Brain Atlas, so that given a new section, we can actually call anatomical regions on it with semantic segmentation. And as a result, we can now apply Tangram to combine single cell profiles with many different spatial modalities: data at lower resolution, like in situ hybridization, intermediate resolution, like spatial transcriptomics with Visium, and high resolution, like MERFISH. And what is remarkable is how many tasks we're going to solve with this one approach or model. First, like with Sarah that I showed you before, we can map the cells into their tissue and we can project their gene expression. We can also correctly predict the expression of genes that were not measured by MERFISH. We can correct raw measurements for genes in methods that have higher quality but more genes. We can transfer other data for spatial coordinates. For example, here we predict the spatial chromatin patterns using RNA expression as the anchor. And when we have both molecular and histological patterns, we can predict the anterior to posterior mapping in order to only map the relevant data and constrain the mapping by the observed number of cells in order to deconvolute multicellular measurements to the single cell level. And when we bring all of these pieces together, we can build one atlas of a complex organ like the brain. We give Tangram our sections and single nucleus RNA-seq profiles in this case, and we get back the anatomical regions, the cellular densities, and the expression profiles of cells and genes in each of the regions. Okay, so now we have static things like types and space. What about dynamic changes like differentiation? We all start with just one, cell, the fertilized egg, and we become this remarkable thing made of 37 trillion different cells. And so in this example, we're looking at early fish embryogenesis that we can monitor along a time course. So several years ago, again, together with Jeff Farrell in Alex Shear's lab, we decided to take a look at this process. We profiled the cells at 12 time points in early development. And the cells that we profiled are color-coded here by the time point from which we collected them. And you can see the progression of time, but you can also see the blending and the asynchrony. By 12 hours after fertilization, there's dozens of different cell types. And so the question is, how did we get from there to here, from one fertilized egg to this enormous variety of different cells? And so for this, we developed another algorithm that we called ERD to reconstruct this process. We start by building a graph between the cells on which we use something called diffusion map, introduced by my colleague Fabian Theis for this problem, to calculate what we call a pseudo time, our prediction of real time, but from unlabeled time points. Then we're going to find a way in order to trace differentiation. And the key is that we're going to start from these endpoints, the tips of the tree, and then we're going to try and make our way back to the root. We do this by a random walk on the graph, but with the important bias that we can only stay at the same pseudo time or go backwards, but never forwards. This guarantees that if we reach a branch point, we can not go down the branch, we can only continue to the root. Now we do this from every tip multiple times. And then if the cells get visited by walks coming from multiple tips, we're going to merge them and we're going to find a tree. And we are finally going to take this tree and lay it out nicely for visual inspection. And so doing this process on our data is going to yield this very detailed tree that I'm showing here, rotating around, and it is colored by the real time point. And it's going to show us the lineages with great resolution. It's going to give us the path for every lineage. And along this path, it's going to show us gene modules that progress across each lineage. And then within each of the paths, we're going to see soon the genes that become active along it. Okay? And so now, again, we would ask ourselves, what kinds of things can we learn from reconstructing this tree? So one thing that we can do now is combine the spatial maps from Sarah with the cell fate maps I just showed you for ERD and ask, how is the fate of the cells at 12 hours related to where they were positioned in space many hours earlier? For example, think about cells like these notochord cells. We can use the tree that we inferred by ERD to trace them back to their progenitors at the same point in time for which we have made earlier the spatial reference map with Sarah. And now when we put both of them together, it can tell us that for these progenitors, where they were spatially many hours ago. And now we can do this from every tip in order to get a full spatial specification map of the embryo. Okay. And so ERD recovered a very high resolution level of biology, but it didn't directly use the time points at which the cells were recovered. This is the case for many of the initial approaches that people developed in this field because they relied usually on single time points and on adult stem cell systems. So in parallel, we decided starting to look at time explicitly. And so for this, we developed another algorithm that we called Waddington OT, which is based on the mathematical approach of optimal transport. And just as a cute aside, this mathematical approach traces to 1780, to Napoleon's army and to a mathematician called Monge. And the goal was to find the most optimal way of building fortifications next to the pyramids in Egypt by moving piles of grains of sand. And so the grains of sand in our case are going to be cells that are going to shift in their distributions. And what is remarkable about this is that while the theory for this existed since the 1780s, the ability to compute on this efficiently only exists since 2013. And so we were able to take this both old and new theory and try to apply it for the first time in the context of biology in order to probabilistically query for cells their likely ancestors and their likely descendants. And in particular, if we look at the population of cells at a given time point, we can ask, what is the likely distribution of descendants from these cells in any subsequent time point? We can also ask, which cells were their likely ancestors at any earlier time point? Or we can take two populations at the same time point, and we can ask, what is their extent of shared ancestry at any earlier time point? And in this way, we can basically play forward or backwards the tape in either directions. And so to see how this would work in a biological system, we turn to a beautiful system developed by Shinya Yamanaka, a distinguished Japanese scientist and Nobel Laureate, the system of iPSC reprogramming. And Jian Xu, a postdoc in Eric Lander's lab, set out to measure a detailed time course in a mouse iPSC reprogramming experiment, collecting cells at half-day intervals over 18 days to a total of 250,000 cells. And now when we arrange the cells on what we call a force-directed layout embedding, or an FLE, and we color them by the time point, we can again see the progress of time, just like we saw it for the fish. And over time, we see that we get a broad range of cells, including the induced pluripotent stem cells, the reprogrammed stem-like cells. And of course, these are the cells we desire the most, and this naturally leads us to ask, can we use the optimal transport model in order to figure out how we get there? And what is special about optimal transport is that it allows us to play the tape backwards from any time point and any population. And so we can trace the ancestors of iPSC cells and see how we pass through this, you just saw it, this very narrow ancestor subset around day 9 and 10. But we can also ask, where do the stroma cells come from? And see that as early as day 2 and 4, after we induce the system, there's already ancestor cells that have a much higher probability of eventually becoming stroma-like cells than any other cell type. Now we can also ask the model questions to tell us what actually drives this process. And so Waddington's landscape is often seen as focused on these intrinsic cell autonomous programs. So we started with those. And we considered in practice a scenario when you can use our transport model to find transcription factors that vary in their expression between cells that are predicted as ancestors of different descendants. We apply this idea to our data, and we look for each cell across each time point here on the x-axis at the likelihood that it is an ancestor of iPSC cells versus other cells by day 18. And even very early on, cells that have a high probability of being an ancestor of iPSC cells express high levels of a transcription factor called OBOX6, about which very little is known. And indeed, if we overexpress OBOX6 together with the Yamanaka factor, the OSKM factors, we obtain a high portion of iPSC. So we can basically predict the future, and then we can manipulate the system to our desires. We can also look for extrinsic factors or cell-cell interactions that drive the process. So for this, we score receptor-ligand interactions between cells of different subsets that occur at the same time. And when we aggregate this across all receptor-ligand pairs, we see that initially cells talk a lot to each other, then this ability is diminished in the first phase of reprogramming, and then it kicks back in dramatically in 2IR serum, and it mostly involves the stroma cells. And so we can use it in order to make specific predictions. For example, the GDF9 that's produced by these stroma-like cells impacts iPSCs that express its receptor TDGF1. And indeed, when Jian added GDF9 daily from day 8, by day 15, there was a substantial increase in iPSCs, but also in neural cells, and this benefits from the same interaction and might compete with iPSCs. Now, the last note I want to make about these dynamic processes is that fate, the ultimate state of the cell, and lineage, the set of cell division events that led to it, are related to each other. But deciphering this kind of relationship can be difficult in practice. Now, when we study model organisms in the last few years, many tracing tools have been combined with single-cell RNA-seq, developed by many different labs. But these methods rely on engineered tools. This means that we can't apply them to human tissues. The alternative in humans would be to do whole genome analyses, which is just very costly and low throughput, and at the end of the day, doesn't actually tell us anything about the cell state. And so Leif Ludwig, a joint postdoc with Vijay Sankaran's lab, together with Caleb LaRue, realized that there was a very compelling alternative in mitochondrial DNA, because first, mitochondrial DNA mutations are known to be a natural tracer, but also mtDNA and mtRNA are captured by single-cell ATAC-seq and single-cell RNA-seq, so we could also get to the cell state. And we often treat these mitochondrial reads as a nuisance, but in fact, they can be gold. And so very briefly, Leif, along with Caleb and Jacob in Vijay's lab, demonstrated this through a series of experiments and analyses. They first showed that we can correctly infer engineered lineages in cell lines. We can recover the same accuracy as a bar-coded cell line experiment. We can recover clones and cell types in primary human HPSCs. In T cells, our mitochondrial clones match with TCR clones. And finally, we can recover clones in patient samples from colon cancer and from CML. Okay, so next I want to turn a little bit from thinking about the cells as our unit to thinking about the programs that the cells use. Because even when cells are of the same type, they can shift some of the genes they use in order to fit their circumstances. And to illustrate this, I'm going to turn to immune cells that are particularly notorious for this. And in particular, I'm going to focus on cells called innate lymphoid cells, or ILCs, which have been implicated in the early steps of psoriasis. These cells that may trigger psoriasis were thought to be ILC3s, a particular subtype of ILCs, but the cells that are present in the healthy skin are not ILC3s, they're ILC2s. So there's an open question of how ILC3s arise to trigger the disease. So we profiled cells along a 4-day time course of induction of psoriasis in a mouse model. But first, let's just look at the cells from the two extremes, untreated, day 0, and day 4. So of course, you can try and group cells, like I showed you before, and at first you might think that looks like a really nice cell clustering. But for many genes, this clustering is actually arbitrarily subdividing the cells, and it does not seem like there is just one way to capture all of the diversity. And to really highlight this point, consider how this cloud in the middle, which is actually where all the action is that comes from both the naive and the treated mice, is distinct from the case of bipolar neurons of the mouse retina, another system that we studied, where each cell type sits in its own tidy and separable cluster. So how can we actually capture this biology? For this, we turn to a computational approach that is used for text analysis. Thinking about cells is analogous to a document, and a gene transcript appearing in the cell is analogous to a word that appears in a document. So imagine that I'm reading a news piece about the latest restaurant of Chef Gordon Ramsay. Hard to imagine these days of the pandemic, but restaurants will come back. And this piece can be related to topics like food and business and celebrity culture, because there will be frequent words used there like dishes names and maybe the amount of money invested in Gordon Ramsay. And the word Gordon Ramsay is actually related both to the food topic and to the celebrity topic. And so in the same way that words in the document result from the topic that the document covers, the gene transcripts in the cell result from the programs or processes in that cell. And so we can capture this by different kinds of computational methods. We rely here on the first one that was proposed for this, called latent Dirichlet allocations. And we will capture the topic as a probability distribution across the genes, where each gene has a weight to belong to the topic, and each topic has a weight to be in the cell. And then this inference is not necessarily always correct, but it structures our data so that we can now come back to it with biological eyes and try to interpret it and functionally validate it. And just consider our day 0 and day 4 ILCs. The topics highlight the different aspects of the cells, not just distinct clusters, like this mixed ILC3s, but also different ways of spanning this big cloud, and even ways in which the program repeats itself in different clusters. And by examining the highly weighted genes, we can start labeling the programs for putative functions. So now we're ready to come back to our full experiment with the time course. If we look at a full time course from naive day 0 mice all the way to naive day 4 mice, we see that the cells actually form a spectrum that spanned almost invariably across all of the days, but that the heaviness across the spectrum is shifting. So in addition to this, we see this spike here, which is nearly, not entirely, but nearly devoid of cells before we treat the mice, but then the cells appear here really rapidly as early as day 1, and then it fills really quickly. And so to characterize this spectrum, we learned a full topic model across all of the cells, and I'm showing you some of the topics, an ILC3-like program, which is highest in this region, and is sparsely populated pre-treatment. And so now we asked, where do these things come from? Maybe those are these important cells for psoriasis. And so we first applied Waddington OT to the data, and we inferred that there is a broad swath of cells across this cloud spectrum that could be ancestors of this ILC3 program. And then we used the ERD program in order to predict the many paths towards this ILC3-like state. And you can get there from the quiescent program directly, or through the ILC2-like program in one or two ways, or from another part of the cloud. And finally, with Richard Loxley's lab, we actually used fate mapping experiments in mice to validate some of these paths. Okay, now that we have these programs in hand, can we actually understand the underlying genetic causes and mechanisms? And we want to do this by using directly genetics as our perturbation in order to have real causal evidence rather than only inferences from observations. And we can do this by relying on either natural genetic variation or by introducing perturbations in an engineered way, and then relying on, in both cases, on the single cell profile as our high content readout. And so I'm going to start with the engineered case and remind us about this beautiful design of the genetic screen. So as a reminder, if we want to find all of the genes that can individually affect the expression of gene X, we would deliver a set of DNA barcoded perturbations to cells such that each cell will receive at most one. We are going to grow them. And then at the end point, we're going to sort the cells by the expression of gene X. We're going to sequence the barcodes and we're going to look for enrichments. This is one of the most amazing designs in biology, but it does have some limitations. And one of them is that we have a very simple readout from each cell, which is the level of expression of just one gene X. And this means that we, first of all, need to know that X is important. We have to choose it in advance ahead of the experiment. And then when we are done with the experiment, all of the hits are going to be the same because they all affect the level of X. So it's going to be hard to capture complex biology. And so a few years ago, we decided to tackle this by devising a method we called perturb-seq, which is a pooled CRISPR screen where we detect one or more perturbing guide RNA and the profile of the perturbed cells by single-cell RNA-seq. One of our first applications was in a cell system known as dendritic cells. This is an immune cell type, which we stimulate with LPS, a component of gram-negative bacteria. And we use this regularized linear model to recover a regulation matrix of the regulatory coefficients of each guide in the columns across each gene in the rows. Blue is the negative effect, red is the positive effect, and the guides and the genes are clustered. Guides with the same target, which are color-coded, grouped together, even though the model did not know it. The genes partition into programs of genes affected in similar ways by the perturbations, and they fit nicely into the biology of dendritic cells. And the perturbed transcription factors, they group into these modules of factors that when we perturb them lead to similar impacts, again, in ways that largely make sense. And we actually learned a great deal of dendritic cell biology from this experiment. But what I want to especially emphasize here is that the presence of these orderly programs makes the perturb-seq assays scalable. When we did power analysis on these first screens, which were saturated for the cells and for the genes, we showed that for a program level effect, it's enough to have as few as 10 to 30 cells per perturbation and a few hundred genes per cell. And so perturb-seq screens, just like these cell atlases, are unifying. We can take one readout and apply to understand effects in vitro or in vivo of coding or non-coding variants for one cell type or for multiple ones at once. And I'm going to show you again a couple of vignettes from this. For example, with Mark Daly, Feng Zhang, and Paolo Arlotta, Xin Jin, a junior fellow at Harvard, used perturb-seq to characterize the function of 35 genes with loss-of-function variants in autism. So this is a case where we know that the gene is causal in the disease, but we don't know which cell or process it participates in, which really blocks us from using our knowledge that the gene is important to the disease towards anything from basic understanding of disease biology or towards therapy. And so we set up a screen where we take a pool of guides and deliver them to embryos in utero, and then we're going to collect the cells and profile them postnatally. And what's important to understand is that instead of making many dozens of mice, each of them manipulated in one way, we can do all of these genes experiment in one mouse at once and look at all of the effects simultaneously. And so when we characterize the effects of the perturbations on major cell types, only one of these 35 tested genes, DIRC1A, had a significant effect on the proportions of major cell types. It increased oligodendrocyte cells and it reduces microglia. But when we turn to the level of programs, everything changes. And so we first identified gene programs across the entire data set. And I'm going to highlight two types of patterns that we see. And I'm using the example of projection neurons. So some programs like PN1 reflect subtype diversity. This is layer 4 projection neurons. And other programs like PN2 are reused across subsets. Here they're highlight neurite development that occurs in some of the cells of each of the kinds. And so when we look at the level of programs, we find that 15 of the 35 ASD genes affect 6 of these programs across 4 cell types. And this highlights the fact that there's likely a limited number of cellular processes that these genes actually converge into. And also the perturbation in one gene can actually impact programs in multiple cell types. So for example, if you look at a gene like ANK2, it induces the NDNF module in inhibitory neurons, but it also affects other modules in astrocytes and in projection neurons. Or if you look at 2 different genes like CHID8 and GATA2B, they both alter the ODC1 module and they alter oligodendrocyte progenitor differentiation, and we validated this in individual experiments. As I told you, we can also use these approaches to look at the impact of natural genetic variation. So in this example, we're looking at the small intestine, and we're looking again at innate lymphoid cells, but those of the small intestine. And we profiled 380,000 cells here across 274 different mice, each of them with a different genetics. They're diversity outbred mice, so they kind of mimic quite well the type of genetic variation that we see in humans. And we use pooling and genetic demultiplexing to make the experiment feasible. And now we can map genetically different features that are affected by the natural genetic variation. We can look at expression QTLs, EQTLs in each of the cell types, but we can also look at effects on cell proportions, and we can even look at trans associations by using cell programs as our traits, and that's the vignette that I'm going to highlight. So this is a program that varies, this is a program that varies across ILC3s and LTIs, which are 2 different kinds of ILCs, and it's enriched for IL-22 pathway genes. IL-22 is a cytokine that is made by ILCs, and it signals to enterocytes, and it's important for gut physiology. One of the 3 genetic loci that are significantly associated with this program harbors a transcription factor called SOX9. It's a key transcription factor, but it's expressed in enterocytes, not in ILCs. So this suggests that there's actually a feedback loop between ILCs and enterocytes. And so the locus might impact ILC cell programs indirectly because of its action on enterocytes. We find multiple examples like this, including QTLs in CGRP, a gene that's encoded by enteric neurons in your gut, and it impacts ILC2s, and that's actually a known axis in the gut that we showed in a different study. And then my last vignette for this part is looking at coding variants, which is what we've done in cancer in collaboration with J.T. Neal and Jesse Bohm. And here we're looking at mutations that we see in cancer patients, and we want to understand what might be their impact on the tumor. And so the idea here is simple. We can distinguish neutral, loss of function, and gain of function variants by comparing the profiles of cells that carry the variants to cells that express the wild type or don't have any overexpression. And this was actually done before with bulk RNA-seq, but that requires individual assays, so it's cumbersome and slow and expensive, and also it only gives us the averages. So we were curious to see if we would uncover more. And so as a proof of concept, we assayed by perturb-seq a pool of barcoded variants in KRAS and in P53, two of the most highly mutated genes in cancer, in KRAS and oncogene, P53, a tumor suppressor. We included as controls synonymous variants and common missense variants from EXAC. We used one cell line, and we analyzed 1,000 cells for variants, which was actually about 30-fold what we would actually need by our current estimates. And at first, we just look at the relationship between the mean expression profiles, this is for KRAS, and we can see that the wild type, which is here, actually groups along with a lot of synonymous controls. And so this suggests this is the wild type phenotype. And indeed, most of the other exact missense mutations, which are prevalent in the human population, map to this cluster as well, although some of them have a distinctive effect, and so they might suggest that there's something else going on with them. Now, the known gain-of-function variants for KRAS group as well, but they group with, and so this is the gain-of-function cluster, and now they group with additional mutations that we can predict are gain-of-function, and finally, there's a few additional mutations with distinct phenotypes. When we compare this to a traditional assay that's a tailored functional assay specifically for KRAS, we see a very nice and clear agreement. We obtain similar results for P53. We have a big loss of function cluster, a dominant negative cluster, and again, agreement with a dedicated functional assay. Now, the phenotypes that we look at are more nuanced, and so the effect sizes from perturb-seq correspond really well to mutation frequencies in cancer. The bigger the effect in perturb-seq, the higher the mutation frequency in cancer. But unlike many other phenotypes, we can now dig into them at the cellular and program level. And so, for example, these KRAS variants, they're not just in discrete groups. They form a continuum, and we can score this continuum by principal components on the single cell space. And when we look at the single cells, we see how we actually move from the neutral to the strongest gain of function. The cells are redistributing in this space rather than occupying a completely new space. It means that even without the mutations, most of these cell states are actually accessible. They are just not used as much by the cells as when the cancer mutation occurs. This only has a weak relation to the cell cycle, but it is related to a distinct expression program, which is more populated by the cells along the gradient of impact from the wild type to the strong gain of function. And there is many, many more such examples, for example, in how we characterize protein complexes like the BAF complex or large families like the E3 ligases. And so finally, I want to go back to where I started, which is common complex disease. And I'm going to go to the example of a form of inflammatory bowel disease called ulcerative colitis. Inflammatory bowel disease is a poster child of human genetics, where genome-wide association studies found hundreds of new risk variants. And they pointed out to new processes and new pathways, and they can give us new therapies. But in most of the cases, we don't actually know the cellular function of the variant, and we often don't know the variant's function or mechanism. And so to try and address this, together with Ramnik Xavier and additional colleagues, we assembled a single cell atlas of 365,000 cells from 12 healthy donors and 18 patients with ulcerative colitis from both inflamed and uninflamed regions. We had replicate sampling. They gave us 68 specimens. And we focus here on a training set of 120,000 cells. And you can see that the cells partitioned to 51 subsets, and they're embedded here in 3 dimensions on the face of the sphere to really help you see the rich variety of cells that exist in your intestines. And they span epithelial cells, where we can recover differentiation, stromal cells, and immune cells. Now that we have this atlas, we can ask, how do things change with disease? And we find that this cellular composition is dramatically remodeled as we move from healthy to uninflamed to inflamed tissue. And it includes some surprises. So, for example, within the epithelial cells, we see these cells that are called M cells, and they're really rare, and they're quite unexpected to exist in the colon. Usually they're in your small intestine, but not in your large intestine. In the stroma, we discovered a new set of inflammatory fibroblasts, also unique to ulcerative colitis. And in T cells, there's a major expansion of CD8 T cells that express IL-17, which is usually a feature of T helper CD4 plus T cells. And so with these rich subsets, we can go back to the human genetic data and ask, in which cells is each GWAS gene expressed. And so in collaboration with Ramnik and Mark Daly, we look at those loci that they already map to the level of an individual gene. And more than 50% of them, which are the genes that I'm showing here, were expressed in specific cell subsets, either irrespective of the disease or only in health or only in inflamed tissue. So now we can turn back to the cells and we can look for subsets that are enriched for the GWAS genes. So for example, these M-like cells that are not even supposed to be in your colon and become preponderant or relatively preponderant in ulcerative colitis, they also have a preponderance of GWAS genes that they specifically express, and also this expression is induced in ulcerative colitis. So now we can ask, how are the cells that are highlighted by this analysis relate to each other in the tissue? So we constructed a graph that connects the cells that are enriched for corresponding receptor-ligand interactions in each of the three conditions. So in the healthy tissue, the key compartments, epithelial, stromal, and immune, are largely delineated from one another. You know, epithelial cells talk to each other, but not so much to the stromal and the immune cells. But in ulcerative colitis tissues, they become decompartmentalized, or all hell breaks loose, and there's many new cross-compartment interactions. And some of the cells that are enriched for the GWAS genes are actually the key hubs of this, decompartmentalization. Okay, so now let me go back to these GWAS genes and ask, how well did we actually account for them? So I already told you a little over half had cell type specificity in their expression, but some of the others do not even seem to have expression in our data set at all. And this can be for multiple reasons, but there's one major one. We don't actually have a full picture of the tissue. And so first, we only profiled the mucosa layer, which is kind of the top layer, and the lamina propria that comes immediately below it in our biopsies. But there are some cells, like fat cells, like adipocytes, that exist only in other layers. And then second, there's a few cell types that we didn't capture at all, especially enteric neurons, which are not just really rare, but also they don't survive dissociation for single-cell RNA-seq. And so we tackled both of these challenges by two new lab methods that we developed called RAISIN-seq to profile nuclei and Miracle-seq for label-free enrichment of rare cells. And we combined these methods and applied them to frozen resections from another 10 individuals. And we built an atlas of these deeper layers, and we recovered very rare cell types like interstitial cells of Cajal and enteric neurons, as well as other cell types like adipocytes. And now that we have this more comprehensive atlas, we take these additional cell types and we end up relating most of the other ulcerative colitis GWAS genes to cells as well, including to specific subsets of enteric neurons, but also, for example, to adipocytes. And we can even identify cell types that are that have specific expression of dozens of risk genes for other disorders of the enteric nervous system, like Hirschsprung's, which is a severe disease of lack of innervation, and also early life and late onset disorders of the central nervous system, but that they have symptoms of gut dysmotility, like autism spectrum disorders and Parkinson's disease. And in some of these cases, the expression in enteric neurons is much higher than in neurons of the central nervous system, suggesting that the gut might be the key locus of gene action. And so this example emphasizes how it's important to have comprehensive catalogs. And so towards this, as part of the Human Cell Atlas that I'll return to in a little bit, we're completing now a cross-tissue data collection, which uses these lab method toolbox that we originally developed for the colon example, but now we're applying it across 8 different tissues, and we can see the extent of both the tissue specificity and the shared cell types, so that only in a full body atlas, we can really address the cell and tissue specificity of disease genes. So now I'm going to take you back again to this analysis in ulcerative colitis, and I'm going to drill into the level of a gene. This gene has the name C1orf106. That kind of means in biology, I don't know what this gene does. I do know that it's expressed in enterocytes, and I know that it's a GWAS gene, so it has a risk, variants in this gene increase your risk of developing ulcerative colitis, but that's kind of it. I don't know its function. I now know it's specifically expressed in enterocytes. How can I predict what the gene does? So one way of predicting gene function is asking with which other genes is my gene expressed. And in the past, we could do this by comparing one cell type against other cell types using the fruit smoothie method or bulk RNA-seq. And if we do it, we would get epithelial genes because C1orf106 is expressed in epithelial cells. But now we can look just across the single enterocytes, the cell type in which the gene is expressed. And we find that within those cells, it co-varies with tight junction genes. So we can postulate that it has a function in tight junctions and the barrier. And indeed, Ramnick's group has shown that it regulates the stability of epithelial adherence junctions. So now we can generalize this approach across all GWAS genes. We relate the genes to cell types. We look for genes that are co-varying within the cell type. And then we iterate this, starting with different GWAS seeds until we converge to a set of modules that connect the GWAS genes and additional ones. And remarkably, most of the modules are enriched from multiple GWAS genes at a time, such that 10 such modules account for more than half of the UC GWAS genes in the same in the cell type specific manner. And now that we know that this structure exists, we could use it in order to nominate risk genes in loci that have multiple genes. By this, I mean that if one characteristic of GWAS genes is that they aggregate together in modules, then can we prefer candidates that co-vary with known GWAS genes or with candidates in other regions? So to test this idea, we took cases where the causal gene is known in a multi-gene region, we hid its identity, and we saw if using either expression or co-variation with genes in other associated regions would help us predict the causal gene. And so we find an almost twofold increased accuracy compared to a null, where we choose the gene at random or by expression alone, but it's only true with ulcerative colitis, the source of our tissue, and not in Crohn's disease, another form of IBD. And also we can now take regions that were not known and predict new causal genes and provide them to the biologists as a prediction. Most recently, we took this a step further in collaboration with Alkes Price to take our gene programs from single-cell RNA-seq data sets with a full genome-wide association signal through a new single-cell LD score regression framework. And when we applied to colon cell type programs in IBD, we found many of the expected relations, for example, for myeloid cells, which are known to be implicated in IBD, but also we found this nice heritability signal in M cells, those cells that we didn't even know existed in the colon. We also found associations to programs that are specific to each cell type in ulcerative colitis. We call these disease programs. And for example, we found a strong signal for a fibroblast disease program. And overall, the disease-specific programs, in particular cell types, had excess heritability for IBD and ulcerative colitis. We see this in epithelial cells, especially in ulcerative colitis, for ILCs, especially in Crohn's, and for T cells in both. Now, you can apply this approach to new diseases too. So, for example, we have done this in the context of COVID-19, a disease that we have studied now in autopsy samples with single-cell genomics from 17 individuals who succumbed to COVID-19, and we had donor autopsy samples to analyze, and we analyzed the lung, the liver, the kidney, and the heart. And we can combine it with the results of genome-wide association studies that are trying to predict genes where variants in our genome increase our risk for developing severe COVID-19. And using this, we were able to suggest cell types that may mediate the heritability of risk of severe COVID-19 disease, including AT2 cells, ciliated cells, and CD8 T cells in the lung, cholangiocytes in the liver, and connected and proximal tubule cells in the kidney. And so all of this brings us full circle, taking the cells of our body to their natural habitat, the tissue, which is critical in the context of human disease. And I'm going to illustrate this with an example from cancer, where we looked at single-cell RNA-seq data that we acquired from 31 patients. Half of them, treatment naive, and half of them treated with immunotherapy, which succeeds in some individuals, but others are non-responders. And focusing on malignant cells, in the top plot, the cells are colored by the patients, and you can see that the tumors are really different from each other, so each patient's cells separate from each other. This is a common pattern we've observed across many tumors, but we can also ask if we can still use it to find common features. And for this, we developed a computational method that combines single-cell RNA-seq from a few patients with bulk RNA-seq from many patients in TCGA. From the single-cell RNA-seq, we first identified genes that are specific to T cells in tumors. We use this T-cell specific signature in order to infer T-cell levels in TCGA. And then we search for genes that are specific to malignant cells in our single-cell RNA-seq data but whose expression in the TCGA tumors correlates positively or negatively with either a high or a low proportion of T cells in bulk tumors. And then finally, we take this seed and we expand it by finding genes that co-vary with the seed across the single malignant cells. This gives us a program that we call the T cell exclusion program. And the repressed part of the program, which is lower in cold tumors and higher in hot tumors, has genes like MHC class 1, interferon gamma, and the SASP. And the induced part, which is higher in cold tumors, has genes like MYC targets, CDK4, and CDK targets. We confirmed that this program is indeed associated with exclusion in situ. We used a multiplex immunofluorescence method together with Peter Sorger to measure 13 proteins in FFPE sections from 19 of our patients, marking the key cell types and the exclusion program. So to orient you, this is one section. In blue are T cells, CD8 T cells are in light blue. In red are cells that are malignant cells that repress the exclusion program, so they should promote a hot environment. Green cells are also malignant cells, but they induce the exclusion program, so they should promote a cold environment. And this is exactly what we see. Red malignant cells are interlaced with the blue T cells, but the green cells make these big cold patches. And this is true across the patient samples, and it can be quantified to show that the exclusion program is significantly higher in malignant cells that are in cold T cell depleted niches. So now we can look how this program behaves in the cells in each of the patient tumors, looking both for variation within a tumor and between the tumors. And when we look at the cells from the untreated and resistant patients, we see that some of the cells from the treatment-naive patients already expressed the program. So it could reflect an intrinsic resistance mechanism that existed before the treatment. And we tested this in a bigger cohort of 112 patients in collaboration with Dick Schadendorf in the University of Essen and Eli Van Ellen at the Dana-Farber, where patients were biopsied before checkpoint inhibitor treatment with bulk RNA-seq and then monitored for outcome. And we found that the signature expression in bulk tumors pre-treatment was predictive of both progression-free survival and of clinical benefit and its duration. And so next we wanted to see if there are drugs that can revert this program. So we queried for drugs that are more toxic to cell lines that overexpress the program than for cell lines of the same tumor type that do not express it. And the third top hit was CDK4/6 inhibitor. Now you might remember that CDK4 and CDK targets were members of the induced side of the program. And so we set to test this prediction. We treated a melanoma cell line with a CDK4 and 6 inhibitor abemaciclib. We profiled with single-cell RNA-seq before and after treatment. Before treatment, about 10% of the cells expressed the program. We see this in purple. After treatment, the proportion is radically reduced. And in addition, the remaining cells induce the downside of the program, especially the senescence-associated secretory phenotype. And then we turn to the B16 melanoma, the B16 melanoma animal model, which is typically resistant to checkpoint inhibitors. And we found that combination therapy, where abemaciclib is added after the initial checkpoint inhibitors, gave us substantial benefit. And this depends on both having RB-competent malignant cells and on the presence of CD8 T cells. And so even with this analysis, we still looked at the programs one cell type at a time. But from a tissue biology perspective, we expect the cells to coordinate their actions, either because they're all responding to a shared signal or because one of the cells responds to a cue and affects the others. And some of this could lead directly to the same response. But even if the cells respond differently, which is more likely, the response should be coordinated. And this means that we expect to see a signal in the data that reflects this coordination. And I'm going to call this the multicellular program. So to relate multicellular programs and find the signal, we need to find some way to relate the cells of different kinds. Being spatially close to each other is one way of doing this, but tissue locations or individuals and disease conditions can allow us to find these patterns from single cell profiles. And we devised a method to look for these signals, which Livnat Jerby, who's just finished her postdoc in my lab, developed as a two-step approach, which we call dialogue. We wanted to test this idea. We went back to the ulcerative colitis atlas. It has 115 spatially distinct samples. And we focused on the 5 well-represented cell subsets: macrophages, transit amplifying cells, 2 kinds of intestinal epithelial cells, and 2 kinds of T cells. And we looked beyond pairwise combinations. And I'm highlighting just one of these multicellular programs. It's made of different genes in each of the 5 different cell types, and they're labeled on the top. But the levels of the genes are coordinated across specific subsets of T cells, epithelial cells, and macrophages. The induction of this program in these cells is associated with ulcerative colitis. It's enriched from multiple genes that are located in risk loci in GWAS of IBD and ulcerative colitis, but these genes are expressed in different cells, but in a coordinated way across the cells. And when we score it with bulk patient RNA-seq profiles, it's actually predictive of the response to anti-TNF therapy. And so I showed you that single-cell profiles hold information on cell type, location, differentiation programs, their underlying mechanisms, and tissue biology, and that they're starting to give us a new vocabulary for disease. I want to finish with 2 lessons. The first lesson is to ask, why would this work at all? Single-cell RNA-seq data is an example of what we used to call bad data. It's noisy, it's sparse, and any given cell may have only a random sample of maybe 5% of the RNA in the cell. How could all of this have actually worked at all? And so to give you an intuitive idea about this, I'm going to use a little example from art. So imagine that doing functional discovery in biology is the same as figuring out which painting is actually hiding in this frame. So every experiment is going to mean uncovering one tile. And I can only uncover some tiles, far fewer than the full number of tiles. So I know something, though, about pictures in the world in general. They have patches of color, which is what I'm interested in, and they have kind of this uninteresting beige background. So until recently, all we could do is remove very few tiles. So we had to focus. So imagine that we already knew from past work that there is a red pixel over here. Because colors come in patches, the best thing that I can do is kind of dig around it. And that's going to allow me to recover a red patch, which is something that I like. Maybe I also know there's yellow here, and then I can dig around the yellow and find a tiny patch here. So I know that there's red and there's yellow. I know a lot about their patches, but I have no idea about the big picture. You know, what if I used an alternative approach? I could just throw these at random at the picture, but almost everything is background. So I kind of got my red and my yellow. I do see that there is a blue pixel, but I know even less than I knew in the other design. But what if I were allowed a lot more tiles, say 5-fold more tiles? Now, as they accrue, even though I just threw them at random in my image, I can actually tell that this is a Miro painting. And if I really like modern art, I also know that it's at the Broad Museum in Los Angeles. Now, this makes perfect sense for people who do machine learning. We actually do it all the time. It also makes perfect sense for our brains, because our brains do this all the time. But doing this in experimental biology was not so typical. Sometimes we do it, sometimes we're not even conscious of the fact that we're doing it. But it's a great new opportunity, and we're already reaping benefits from it. And that is what single-cell RNA-seq is about. And so in our very first single-cell studies, which were in dendritic cells, we saw 2 important things. Expression within cells and patterns across cells are highly structured, just like that painting. And also, you know, our sampling of the transcripts, it's really sparse. And from this, we made a major and conscious experimental design decision. We will develop methods like DropSeq that I mentioned in the beginning, which favor sparse, noisy data from massive numbers of cells over richer, precise data from small numbers of cells. We have this structure of gene expression, and we'll make sure that we can handle the data. And that's exactly what happened. I call this line of thinking design for inference. It was very uncomfortable at first, but it actually works. The second lesson, and very clear one, is that having a map is so much better than not having one. And it's so useful. Why not make an atlas of all human cells? And so this led us to start the Human Cell Atlas Initiative, whose mission is to create a comprehensive reference map of the types and properties of all human cells, the fundamental unit of life, as a basis for understanding, diagnosing, monitoring, and treating in health and disease. We sometimes call it fondly building a periodic table of our cells, but this is not just a metaphor. It actually conveys our hope that from this data will not just arise a catalog of cells, but our real understanding of the theory of the cells and our ability to predict them and understand them better. Also, humans and their cells are diverse, and scientists are diverse. And so this should not be something that any one lab, one institute, or even one country does alone. And so that is how the International Human Cell Atlas Initiative became an open initiative, which is a rapidly growing community open to anyone who is willing to adhere to its mission and values. We have over 2,000 members now from 76 countries and over 1,200 institutions. And it has a very large and pioneering presence in Japan since before its inception. We're charting more than 15 major organs and systems with scientists organized in biological networks. The networks work together, collaborating across labs, institutions, and countries. And for example, the Human Cell Atlas Lung Biological Network has come together in January and February of this year during the beginning of the pandemic to really understand how SARS-CoV-2 virus might infect our cells. We learned a host of invaluable lessons from the Atlas. I gave you a glimpse on one of the latest studies earlier on, and we have extended them to COVID-19 patient samples, looking at bloods, aspirates, nasal swabs, and even organs from autopsies. Together, we're mapping the entire human body. We have collected millions of cells from hundreds of individuals across the world for each of our major organs. They're already leading to valuable insights, and now the ability to really query this atlas. These are only the early days for the Human Cell Atlas, but we're well on our way to hit our first draft at the 5-year mark and work for a comprehensive atlas of up to 10 billion cells in our decade-long effort. And with this, I will conclude, and thank you again so very much for this wonderful honor.
Thank you very much for the excellent talk. So I'm very pleased to hear your talk. So as we have just heard from her, so single-cell genomics has revolutionized our understanding of many aspects of biology, revealing new cell types and their interactions, and importantly pointing the way to new, more effective therapies based on stronger and deeper understanding of the composition of the human body. So now let's proceed to a Q&A section. So if you have any questions, please use raise hand button on your PC.
Dr. Sik Du Tan, please. Now you can question.
Hi, thank you for the very interesting talk. Have you looked into the mesenchymal lineage for the actually, I'm very interested to look into the mesenchymal lineage, especially for the tendon formation in the early development. So I'm not sure if you have any data related to this that possible for collaboration in this aspect. And the other thing I would like to ask is about the COVID sample that you mentioned that you have analyzed from the biopsy. So are these from the postmortem biopsy sample? And what is the challenges of using the biopsy sample in the single cell sequencing analysis? Okay, thank you.
Great. Okay, so there were two questions. I think I heard correctly that you asked about mesenchymal cells and differentiation in the context of the tendon. Am I correct?
Yeah. I'm working on the mesenchymal stem cell differentiation. But in the earlier part of your presentation, you mentioned about the development analysis that you use the zebrafish at the different development stage, if I'm correct.
Yeah, absolutely. I wasn't sure about the mesenchymal stem cells in which organ. So I'll give a general answer, and if I miss something, just tell me. It's the audio is not perfect for me. So first of all, absolutely, you can infer a great deal about mesenchymal stem cell differentiation. We had a study on this in the context of the bone marrow stroma with David Skadden, actually published maybe about a year and a half ago, that I think also recalibrated a little bit some of our assumptions on the mesenchyme, on the stroma in the context of the bone marrow, and that is one of the most heavily investigated stromas. I would say in general for the stroma and for stromal-related studies, single-cell RNA-seq is proven to be extremely useful. And this is because, as you probably know better than I do, the ability to actually sort cells with the right markers, say for fibroblasts, has not been the easiest endeavor in the past, which made enrichment difficult, because when you try to enrich cells, you might have gotten kind of the wrong subset, either only a portion of the cells or a mixture with other cells. So you either missed some or had some unintended visitors, and you would never know that. But with single-cell RNA-seq, you can just look at all of them. And we make a lot of new discoveries, especially around fibroblasts and the processes of differentiation, and also new cell states for fibroblasts. And more generally for the mesenchyme, also for other stromal cells, endothelial cell biology gets really enhanced in this way, and so on. The second is specifically for fibroblasts, the ability to culture them and maintain their kind of shape-shifting nature is difficult, because once you put them on plastic, they tend to often lose a lot of their unique characteristics in the tissue. So this work directly from the tissue is greatly enhanced. If you do actually study the tendon, and I heard that correctly, then I want to point out that actually in the Human Cell Atlas, as one of the seed networks, there is a network that actually focuses on the tendon. And I would be happy, in an email, I would be happy to even make the connection and highlight it. You can always also go on our website and see all the biological network, and each network has actually an email address that goes to the 2, 3, 4 biological leads of that network that can always add people and connect them to these activities. The second question was on the autopsy samples, and I actually am glad to be asked, because I put it a little bit as a plug so somebody would ask me. So this was, first of all, a heroic effort of many people. This specific study is from the Boston area hospitals, Beth Israel, Brigham, and Mass General, but it's part of multiple studies that have happened in multiple places around the world as the pandemic hit our own backyard. So there's a big study also from Columbia University, led by Ben Izar. There's a study from Cornell Weill, also in New York. There's a lot in the US from New York and Boston, because we, of course, were communities that were hard hit early on in the pandemic. There are studies from the West Coast, from Stanford. There's work from the United Kingdom, work in Germany, and work in many other countries. But it really sadly tracks, of course, together with the pandemic. This was a major effort because the tissue, as it comes out, of course, from patients, is infectious. It has to be handled in a BL3 facility. We had to develop a unique pipeline for that. Our scientists had to train to work inside spacesuits to do single-cell genomics, but they did. And in particular, we relied on the method called single-nucleus RNA-seq that I already mentioned, because it can be applied to frozen tissue. So we could first separate the tissue and freeze it, and get much better outcome out of autopsy tissue than when you try to take autopsy tissue that already sat sometimes for 24 hours before the autopsy was conducted, and try to derive, a viable cell suspension and do single-cell RNA-seq. So it relied mostly on single-nucleus RNA-seq. There were computational challenges. These autopsies were long-term. These autopsies were had long, what is called post-mortem interval of, as I said, 24 hours and sometimes longer for some of the autopsies, which means some of the cells were dying, which means the RNA spills out, and that can contaminate. Later on, it's what we call ambient RNA. We used an algorithm developed by one of my colleagues to clean ambient RNA. It had a big positive impact. And it required us to almost do like a little human cell atlas, but in a few weeks, because we actually didn't have just 4 different organs. These are the first 4. There's another 8 or so following on them, which means we had tissues from many places in the body, and we had to compare them to references. We had to come up with a way of doing the comparison to the healthy reference in the Human Cell Atlas to derive the annotations very quickly and to start looking at patterns. So it was a huge effort of many, many labs and scientists, and it's been a great privilege to participate. We feel that we made some important findings there, both in generating the resource for the community, which is going to be open very soon in processed form, but also in seeing, for example, some important analogies between the severe COVID-19 in the lung and features, for example, of lung fibrosis, and other impact on both the immune cell compartment and the epithelial and stromal compartment. So we hope will help physicians as they think about treatments, both in new drug developments and applications of existing drugs. More generally, I would say, I think I made that point. In single cell genomics, you have to adapt your methods to the problem, not the problem to the method. I think it's a fearless field in trying to always come up with more experimental innovations and more computational innovations, and they go hand in hand. And in handling autopsies, working with frozen material and nuclei has been one of the greatest boosts that we had in really getting to the depth of some diseases.
Second question, that for the biopsy sample, because in the molecular work, we know that the RNA are easily degraded. So I'm not sure for the autopsy samples, so the single cell RNA sequencing, the total RNA extracted from these autopsy samples for the single cell sequencing, would the RNA integrity difficult issues for these type of samples? Thank you.
It's a great question. It's always better to have fresh and immediate over sat for a long time. But the question is, what do you do when you have no choice? And the COVID-19 study, there was no choice. This is what it is. Now deal. And I think that's what's important, especially in conditions like the pandemic. You no longer get to choose your experiment. You no longer get to set it in ideal conditions. Can you still learn valuable biology? The answer is you can. And the answer to that is made of two components. The first is, as I pointed out, studying nuclei was a big boost for us. There's sufficient high quality nuclei with good RNA in them. And we know that it's good RNA because, in fact, there was reproducibility across the individuals. There was reproducibility across studies conducted in different cities. There was agreement between data that we had from short PMIs and long PMI postmortem interval and long PMIs. So while we would have preferred other samples, we would have honestly preferred no samples and no patients with COVID-19. But under the circumstances of what we could do, we found that the data was okay. The biggest hurdle that you have is cell type specificity because of ambient RNA. And this is where the second component kicks in, which is the ability to use computational algorithms to address challenges. The process that generates errors or noise in single-cell RNA-seq can actually be modeled. Both the experimental limitations of the molecular biology, if you use droplets of the droplet process, of dead cells spewing out RNA that becomes ambient RNA and reduces the quality of your data. And that has been modeled by a generative model developed by Mertash Babadi called CellBender. And it's been a godsend, or a Mertash-send, in the sense that it really allows us to clean a lot of the noisy signal. And then finally, we need additional computational algorithms to do the analysis to relate this data to the data that we have for much more pristine conditions, for example, for the healthy lung. And that is part of what gives us the confidence that what we're seeing is actually biology rather than experimental errors. So it's not perfect, but it's actually, with a lot of determination, there's a lot that can be learned under these kinds of circumstances.
As limited, so we would like to move to another question. So Miyawaki Sensei, please.
So this is Atsushi. Thank you very much for your very beautiful talk. And I was very much impressed by the power of optimal transport. And I like lesson 1, so especially the design for inference. But I have a very naive question, because highly differentiated neurons do have many thin and long processes, including dendrites and axons. And RNAs go into those processes for local protein synthesis, right? So during the dissection or disassembly process, so we may lose some RNAs. And in our experiment, we have an opinion that such spatial pattern can contribute to cell-to-cell heterogeneity. So can I have your perspective from a technical viewpoint?
Yeah, it's like a planted questions where you really hope somebody would ask you something and then they do.
Right.
So glad you asked. So I'll start by saying that the nature of neurons, especially adult neurons, and the older the brain, the bigger the problem, actually made them the worst original target cell for single cell RNA-seq, because at some point the brain becomes undissociable. You simply can't. And the source, the reason we developed single nucleus RNA-seq, which was done by Naomi Habib and Yinxing Li, together with Feng Zhang and my lab, so it was a collaboration between my lab and Feng's lab, way back when, was that we wanted to study neurons together, and we couldn't do it with dissociation, so we turned to develop the single nucleus RNA-seq method. I think there was a lot of skepticism in the beginning, and I don't think unjustified. It was another example of something where you can't do the other thing, so you kind of feel like you're settling for less. And what is interesting that has happened in the neuroscience community is that after a couple of years, it became extremely embraced as an approach. Because for the first layer of the problem, just knowing which kinds of neurons are there, single nucleus RNA-seq is actually a really effective method. And even if you think about things like, activity-dependent behavior, which tends to involve new transcription, and I'll get in a second to the projections, they're going to come back, just to be clear. But for activity-dependent transcription, which depends on new transcription, the nucleus is a great place for it, because you can distinguish the pre-mRNA from the mRNA in a method that was developed by Peter Karchenko and Stan Linarson called RNA velocity. You can even predict the future of the cell from looking at the nuclei. You're saying the RNA, the mature RNA is the current state, the pre-mRNA is the future state, so you can even look at dynamic processes. These are all great things that studying neuron nuclei gave us. But they don't give us the full story. They miss at least two critical things for us in the brain. The first is what you've just said, that the neurons are one of the primary examples of non-uniform distribution of RNA inside the cytoplasm in the projections, and for local, for example, translation of RNA and other RNA regulatory mechanisms to actually control how cells synapse and so on. And the second layer, of course, that a lot of neuron function is in the circuitry that they form. The projections go far, far away from where the cell body is, and you want to find those relationships as well. And this is where actually the marriage of single cell genomics and spatial genomics really gets highlighted. And I showed the algorithmic side of it, but I didn't actually talk about the biological side of it. So from a technical perspective, what this gives you is the ability to actually toggle back and forth and end up having your cake and eating it too. So you can start with a single cell information, single nucleus RNA-seq, for example, to know the spectrum of cells that you have in the tissue. Identify the set of probes that would be the most informative, right? The best set of probes that you might want in order to characterize that many different cells in a tissue with, say, a method that gives you 150 probes like MERFISH. As an aside, you can use compressive sensing methods that I didn't talk about, that lets you use 150 measurement channels and end up with maybe 1,000 or 10,000 genes. So there's kind of a future trajectory there to even go better. But at minimum, to choose your probe. Now you do your spatial measurement, and with the spatial measurement, you can do a lot of amazing things. You can look at projections, you can see the distribution in the structure of the neuron, but of course you only have 150 transcripts. So this is where the algorithm comes back, and you take the cellular profiles that you have only from the nuclei, you map the nuclei back to the space, as I showed you we have done actually for MERFISH, and you project the remaining, 19,500 and something genes in the genome. And by iterating this process, you can actually learn and have both a genome-wide view of the tissue and a spatial understanding of it. I think for this, you also need higher resolution imaging that lets you go subcellularly, because it is quite likely that there is a model of that subcellular distribution that we can learn as well. But that's just an assumption, not based on data. I just can hardly imagine that is not possible given how reproducible it is.
Thank you. I like spatial genomics, and I think spatial genomics is to some extent dependent on nuclear genomics. So you can infer spatial pattern of gene expression from nuclear RNA in the future, and that entirely depends. But I'm very looking forward to it.
Yeah.
Thank you.
Thank you.
Okay, so the last question, Iwasaki-san, please. Question now.
Okay, can you hear me?
Yes.
Okay, great. So thank you very much for your lovely talk. And I was wondering if it is possible to combine single cell analysis data from different species and analyze them from the evolutional point of view. So since you have got very impressive data from cell fate or developmental analysis, I thought that cross-species comparison may enable us to understand very fundamental part of the regulation.
So the answer is absolutely yes. We have actually done it. We've done studies like these in the retina. I did not talk about our retina work. That's actually our first demonstration of a big atlas, was done in the mouse retina. And we have studied them all together with Joshua Sains, a great retinal biologist. We have done the macaque retina and the human retina, and have done a comparative analysis of mouse, human, and macaque. And together with Josh, especially Karthik Shekhar, who was a postdoc in my lab and now has his own lab, they've extended that. We've extended to a full phylogeny, right? Like a much more extensive phylogeny that allows us to look at those things. And you learn really interesting things. For example, for some subsets of neurons in the retina, as has also been shown, Ed Lin has had beautiful work like that on cortical neurons, comparing human and mouse. And now there's human, mouse, macaque, and it's another non-human primate, marmosets. There is extreme high level of conservation. It's like to the point that it's really often one-to-one mapping, and you can really say these are those. We have very recently, this is not published yet, but we have very recently used the Tangram algorithm that I showed you that we use for the spatial mapping. We used in order to take neurons from one species and predict a spatial map in that species based on the spatial organization of the other species, which is something that we can, if you don't have all the data in all species, you can also do. But we also found in the retina cases in RGCs, in retinal ganglion cells, where both the proportional distributions are very different. So where you would have, for example, in mouse, you would have a lot of different subsets, all of them equivalently rare, whereas in human, you would see a few much more dominant subsets and the more skewed distributions of proportion. And also in terms of mapping the subsets of cells to each other, there were subsets that we couldn't actually map in terms of their marker genes. Like you would have kind of a similar number of cells in similar categories, but you couldn't actually say this goes together with that, which is different than the cortical neurons of human and mouse for some of the retinal neuron subsets. And what was particularly interesting was that the transcriptional code, like which transcription factors are expressed in which transcription factors get co-expressed together, that actually seemed to be conserved. So it was as though you had a code to make a certain diversity of neurons in terms of number of transcription factors you need and their identities, but they activated different genes in order to give the cell identity programs. That's one of these interesting things that we're still wrapping our head around, a couple of years after publishing the paper on that. And there are groups, although that is not mine, there are groups who do very extensive phylogenetic work now for cell type relationships. And that goes into the question of how do cell types evolve when you move from a system that is, say, lower diversity of cell types to higher diversity of cell types in the same system. Where do they come from? We have looked at that a little bit in thinking about cells also across different epithelial tissues, and what kind of the complement of different kinds of cells that you would have in every epithelium and would be conserved across species, and things that are more specialization. And then finally, former postdoc of mine, Jet Schibinger, works on the problem of comparing. He actually developed optimal transport, which I showed, he brought the math of optimal transport into this problem of cell differentiation. He's a mathematician and statistician. And he works on the question of if you have two dynamic processes,
So say you have a reconstruction of differentiation in one species and another species, how do you compare that? Which is, again, a fantastically interesting question. I think one of the good things this does for us in terms of cross-species comparisons is that we used to be extremely dependent on markers. And for a lot of systems, the markers, the things that are on the cell surface and that we sort cells by, they're the most fast evolving part of cells. So especially if you went to a system that diversifies quickly, like the immune system, the thing that diversifies the most is kind of this exterior of the cell. That's a well-known thing in molecular evolution. But of course, that makes it really difficult to say this cell in the human is that cell in the mouse. Whereas with these more agnostic, unsupervised approaches that we apply in single-cell genomics, we first say, this is the trachea of the human, and this is the trachea of the mouse. First, let's look at everything, and then let's use these high, these approaches, these embedding approaches, to actually map, learn to share a shared space, and so on. This is also an area that machine learning is finally getting really, really good at. So it's very good times for that kind of problem. Thank you very much.
Okay, so now it's very good time to close the discussion section. So thank you very much again, Dr. Regev, for your really fantastic lecture. Thank you.
Thank you very much.
Okay, now it's time to make a concluding remark. So my name is Yoshiaki Kubota from Keio University School of Medicine, committee member of this prize. And it's my very really great pleasure to make a closing remark for the Keio Medical Science Prize lecture 2020. It's really my pity that we could not have this amazing lecture in person here, but I believe everybody gets many things from this lecture for your own research and medical practice. So, and I hope Dr. Regev keeps in touch with us for future collaboration and mentoring. Okay, thank you very much again, Dr. Regev, for your really fantastic talk. So I'm going to close this symposium. Thank you very much.
Thank you so much.